I will start with the first lesson from Unit 1. This lesson is about water. So the part of the syllabus that we'll be covering today, it says understand the importance of water as a solvent in transport, including its dipole nature. So we'll be talking about the nature of water, and we'll talk about its role as being a good solvent. So first of all, we'll look at the structure of the water molecule. There is something special about the arrangement of electrons in this formula here. So you're probably familiar that there are two electrons here. There are shared electrons in the water molecule. Now, oxygen, being like a larger atom, pulls those electrons to its side. Now, since electrons are negative, then this side of the molecule becomes more negative than the hydrogen sides. And we describe this oxygen as being partially negative, while the hydrogens are going to be partially positive. We have actually a symbol to represent this partial negative and positive charge. We use this small delta sign for partial negative or partial positive. So having two poles within the same molecule makes it, so di here means two, so it makes water dipole. Di means having two poles, a partial negative pole and a partial positive pole. So that's the way the water molecule is arranged. You have slight negative and slight positive charges within the same formula. Now, if you have two dipoles right next to each other, like here, then the hydrogen in one water molecule will attract the oxygen in the other. So again, since this hydrogen right here is partially positive and this is slightly negative, then they'll attract. Positive and negative, you know that they attract. This weak attraction that we have here is what we call hydrogen bond. That's because it involves always hydrogen. These forces of attraction are quite weak, but they're really important for all of the properties that the water molecule has. In this picture, we see many water molecules. They're all attracted to each other. This attraction is very important because it makes all of the water molecules cram together, and this is known as cohesion. So you see that the water molecule in the middle has actually made 4 hydrogen bonds, and it takes a lot of energy to break those bonds, as we're going to see later on. So right here we see how water molecules attract to each other when they are inside vessels. So this picture is taken from a plant stem. You see how the waters are attracted, and this creates like a stream of water molecules. They're all attracted to each other. This is known as cohesion, but they're also attracted to the wall of the vessel here. So the attraction to the wall is called adhesion, while the attraction between the water molecules by hydrogen bonds, it's called cohesion. One property of water that really depends on hydrogen bonding is the fact that it has really high specific heat capacity. It takes water a lot of energy to break all of these hydrogen bonds that we have seen. So this is known as specific heat capacity, which is how much heat it takes to raise the temperature of 1 liter of water by 1 degree. So again, if you've been asked, why does water have really high specific heat capacity compared to other liquids? You're going to have to say, because this heat here in the kettle is required to break those hydrogen bonds that exist between the water molecule. You really need the definition of specific heat capacity. It is the heat needed to raise the temperature of 1 kilogram or 1 liter of water by 1 degree. Another property here is called latent heat. What is latent heat? Latent heat is the heat needed to turn the water from the liquid state to the gaseous state. Latent heat is also high exactly for the same reason. Latent heat is high because there is heat needed to break those hydrogen bonds. So just make the difference here between latent heat and specific heat capacity. So latent heat is the one needed to change state. So that is heat required to change state. Specific heat capacity is the heat needed to raise the temperature of water by 1 degrees Celsius. So that's called specific heat capacity. Here we have two beakers, and you can see here the heat is absorbed by the water molecules to break the hydrogen bonds and change their state. So that is what we call latent heat. It's really important for life on Earth that water has high latent heat, because otherwise all of the water on our planet would have evaporated. So having high latent heat has maintained life on our planet, because this keeps water being a liquid. So all of these aquatic animals like fish wouldn't have survived if water had lower latent heat. Now this is the part that's really important in the syllabus. You need to explain why water is such a very good solvent. So if you drop like a piece of salt into water, so something like this, as soon as the salt is dropped in water, you realize here that the ions get surrounded by the water molecule. So you probably know that ionic compounds have like positive and negative ions. Now take a look at the way the water molecules are arranged here. The hydrogens point to the negative chloride, and the oxygen points to the positive sodium. This results in the formation of what we call a hydration shells. So you see these circles surrounding the opposite ions. They insulate these ions and they prevent them from attracting to each other. So these are called hydration shells. So that's what happens when you dissolve something that is ionic into water. This also applies to sugar. So every time you dissolve something that is polar or charged, then these hydration shells ensure that the things you've dissolved is going to actually be dissolved. We're going to practice what we've just learned about water using this passage. This is taken from an exam paper. So they want us here just to write basic information about water by filling in the blank. So first line says water has the chemical formula. So I'll just write the formula here. And described as, so the only description we've seen today is dipole. Alright, and then they said as a result, individual molecules form bonds with each other. So what's the name of the bonds that water molecules form with each other? These are called hydrogen bonds. Water is important because most biochemical reactions take place in aqueous solutions. So what is the property of water that makes it a good solution? So we say it's an important solvent. Water also has a high, which means that its temperature remains relatively stable despite largest changes of temperature in the surrounding environment. So I'm going to have to say here specific heat capacity. So specific heat capacity. Right, so I wrote specific heat capacity here because they're talking about changes of temperature. They didn't specify anything about changes of state. We'll move now to the second part of the syllabus that we're going to be doing today. Here they want us to talk about carbohydrates. They want us to talk about monosaccharides, disaccharides, and polysaccharides. So first of all, we'll define what a carbohydrate is. So a carbohydrate is just any molecule that is made up of carbon, hydrogen, oxygen. Now, the way these carbons, hydrogen, oxygens are arranged, and the ratio of these atoms makes carbohydrates special. So in general, carbohydrates could be divided into those that are soluble, those like you find in fruits like fructose, or those that are insoluble, like the ones you have in pasta and bread. These are starch. You also have carbohydrates that are not edible. Those are like more of structural carbohydrates, like cellulose, the one you have in ropes or cell walls. So let's start with monosaccharides. Monosaccharides are like the building blocks, the monomers of all other carbohydrates. They are usually soluble, and they have this specific empirical or general formula, which is CH2O. So carbohydrate is a monosaccharide when it has this specific formula. So this is more like the formula of water here, because you see there is H2O with a carbon. So that's where the name carbohydrate came from. So it's carbon because you got this carbon, hydrate because the ratio of hydrogen to oxygen is this one. So only monosaccharides actually have this specific or unique general formula. Monosaccharides fall into many categories. We're only concerned about two of them. One of them is called hexoses. A hexose, hexa means 6, is a carbohydrate that has 6 carbons. There's another one called pentoses. Pentoses are the ones you find in DNA. We're not going to be talking about these ones today, but just to know the term. So we're mostly concerned today about hexoses. One very well-known hexose is glucose. These are notes taken from my book. It's like gives you brief summary of all what we've said today. So don't forget the ratio, the CH2O, so 1 to 1. And there's also the two categories that we've mentioned right here, the pentoses and hexoses. Now we're going to move to the part about glucose. Now, look what it says. It says that glucose has 2 forms or 2 isomers. One of them is called alpha and one of them is called beta. So let's study those 2 forms of glucose. So let's start with alpha. Now, as you can see here, the carbons and hydrogens are arranged in a ring structure. This ring structure is the form of glucose that we need to be familiarized with. So I really advise you here to study or even memorize this structure. Now, you could see that the carbons, there are like 6 of them here. So you start from this, you count clockwise until you get to 4, 5, 6 carbon. Now, as we said, glucose has 2 formulas or 2 forms. It could be alpha. In alpha glucose, the OH on the first carbon is pointing down. In beta glucose, the OH is pointing up. Most of the glucose molecules that we're going to be studying here is alpha glucose. One structure that has beta glucose, which is the only one we're concerned about here, is cellulose. So all of the things we study are basically made up of alpha glucose. So again, just familiarize yourself with the structure and know the unique form of glucose that you have, the alpha and the beta, and they're different by just the position of the OH in their first carbon. So glucoses can get attached to each other. So monosaccharides in general can attach to each other and form a bond. Now you see here, this is like alpha glucose and this is another alpha glucose. How will I know? Because of the position of the OH here. Now when you have those 2 glucoses next to each other, they can actually bond. The bonding here forms by removal of a water. So you see here, we took away a water molecule and we ended up forming this bond. This bond is called glycosidic. And the result here, you can see it's a disaccharide. Di is 2, so we've bonded 2 monosaccharides or 2 glucoses together. We ended up having a disaccharide. So one more time, right at this moment, we got the 2 alpha glucoses. I'll number the carbons, so 1, 2, 3, 4, 5, 6. And here's the same thing, 2, 3, 4, 5, 6. Now you see that this OH here and this OH here, they can actually condense together. That's why the reaction is called condensation. And we could actually take away this OH and this H. This will remove a water molecule. Now the result here is a bond, so there will be a bond as a result here. The bond is like this. There's just an oxygen in between those 2 glucoses together. After that, we've removed all of this here. This bond is called glycosidic. Now it's really to tell the examiner, if you're writing this in exam, you really need to tell them where is this bond. So the bond is between the first in this glucose, the 4th in the other glucose, right? So it's 1,4 glycosidic bond. And the reaction is called condensation because we removed a water molecule. So these are things you need to memorize. You need to know like what are the 3 disaccharides and what are their components. So sucrose is the one found in fruits. It's a combination of glucose and fructose. Lactose is the sugar of milk, is glucose and another monosaccharide called galactose. And maltose is condensation reaction between 2 glucoses. So these are 3 disaccharides that we really need here for our syllabus. So here's an exam question. This is taken from my past paper book. It's about comparison between alpha glucose and galactose. So it said, use the information in the diagram, explain why these 2 molecules are classed as monosaccharides. So what makes those in the picture here monosaccharides? So the unique properties of monosaccharides are, first of all, the fact that they have this ratio. So if you had the time, we'll count the carbon, hydrogen, oxygen. You realize they have this ratio. Another thing is that they are not disaccharides because I don't see any glycosidic bond, right? So each one here is just made of 1 unit. Then they said, use information, find 2 differences between those 2 molecules. So what are the 2 differences we see here between those 2 molecules? So one clear difference here is the position of this OH, right? So that carbon 1, the OH is like flipped. Another difference I see on carbon number 4, again the OH is flipped. So you just need to tell them like what are the differences we see here in these 2 molecules, and you score those 2 marks of the question. Finally, they asked me here for multiple choice question. They said molecule of alpha glucose and a molecule of galactose joined together, what we call the disaccharides. Disaccharide is the milk sugar, or if it's lactose. Describe how 2 monosaccharides join to form a carbohydrate. So they want me to write it up. How do monosaccharides join to form lactose? So in general, the name of the bond is called glycosidic. You need to tell them like what the name of the reaction. Since the reaction involves the removal of water, is called condensation. And you need to tell them like where is the water molecules been removed? It's been removed from between the 1st carbon and the 4th carbon. This table is taken from my book. It shows you the differences between the condensation reaction, the ones we've been talking about, and hydrolysis reactions. So condensation involves making bonds. Hydrolysis, we break down bonds. Good example of hydrolysis reaction is actually digestion. So when we digest, we add water to break bonds. When we are building up larger molecules like disaccharides or starch polysaccharide, then we actually do condensation reactions. Finally, here they want me to draw a lactose molecule. So how am I supposed to draw this? I need just to copy the monosaccharides. So I copy these here. And as I've done earlier, you need to remove a water molecule, and then you show the bond. The glycosidic bond always looks like this. So again, copy these, show the bond, and show them here that you've actually removed a water molecule. So 3 marks, 1 for the bond, 1 for the water molecule, and 1 for you just copying these structures. So this is taken from the marking scheme. It shows you how they've actually drawn the glycosidic bond.